Hi, Dennis. Welcome to the podcast.
Very pleased to talk to you, Jitendra.
So, of course, I was thinking about this question: what drives evolution, and how we can start the conversation in a way, but as being a biochemist and molecular biologist, I reached to that point, to kind of understand what is the code of life, what is that message. So main kind of notion in the field is that it is the DNA, it is genome, which is the code of life. So what are your views on this kind of notion of the code of life, and especially DNA being at the center of the whole theater?
Very, very good and very wide question. And what I'm going to say does not in any way whatsoever challenge the major discoveries of molecular biology in the 1950s and 1960s, where the work of Watson and Crick and dear me, I've just forgotten her name now. The lady who got that beautiful
Rosalind Franklin.
Rosalind Franklin, there we've got it. When I mentioned her in a big debate with Richard Dawkins last year, there was huge enthusiasm from all the women in the audience, but a few men were applauding too.
Yeah.
Yes, Rosalind Franklin. No, I don't challenge in any way the great significance of the discovery of the double helical nature of DNA and the way it is arranged in cells and functions in cells, nor do I challenge in any way the one-way sequence coding, if one wants to use that word, from DNA to RNA to proteins, and no possibility of the sequence of amino acids in a protein somehow being back translated into RNA or DNA. And indeed, it couldn't be, for very good technical reason. The coding between those DNA sequences and the protein amino acid sequences is redundant, and therefore it's actually, strictly speaking, impossible to go the other way, using the sequence of amino acids to get the exact sequence of the nucleotides. But I maintain that that's not the way in which we should expect an organism to try to direct its evolutionary reaction to changes in the environment, because control mechanisms are present. Now, why are they present and how are they present? And here we just have to be a little bit technical for a moment on the exact molecular biology and the process of control of the molecular biology. Usually, the strict reductionists would say that that central dogma of molecular biology, from DNA coding to protein amino acid sequences, is never reversed. And that is true. However, that is not the way in which organisms control what happens. And that arises because of a very simple fact, which is that the DNA is not itself a self-replicator. It replicates, it gets replicated, and I think there's a better expression for it, but it's not a self-replicator. And let's just go through the detailed molecular biology. The double helix, as everybody appreciated when the central dogma was formulated, means that each nucleotide naturally connects to its partner. The CG80 coding is based on that. So when the double helix unravels, as it must during copying and replication, each of the nucleotides that like to be together attract each other. That's a simple chemical fact. So as you have one thread of the double helix open, the appropriate partners will naturally, chemically come in place. Now, that's very much like crystallization. Crystals form and grow because the molecules in solution, not yet in the crystal, find it easy in a kind of lock and key fashion to fit into the rest of the crystal. And that's how a crystal grows. Now, that is partly true of how DNA replicates, because as the double helix is unraveled, each of the nucleotides will bind up chemically with its partner. However, there's always an energy to that process of binding. And that means from that energy of binding, you can calculate how often that will go wrong, or you can just simply measure how often it goes wrong. We know the answer to that. It's about 1 in 10,000 base pairs. Now, that is remarkable if you think about it. Before we go on to why it's not sufficient, it's remarkable because if you or I wrote a 10,000 word article and we had only one typo in that 10,000 word article, we would be very pleased. So would the publisher. One tiny correction to do, that's the end of it. However, that is sufficient to come back to nature, to life. That is sufficient for small viruses. A typical coronavirus of maybe 20 or 30,000 base pairs, well, it will get the odd 1 or 2 changes that occur during each replication. That's incidentally why viruses mutate fairly quickly. So during the coronavirus pandemic, we've seen that happening before our eyes. However, your and my DNA is 3 billion base pairs long. Now, if you do the calculation, the difference between 10 to the 4, 10 to the power 4, which is 10,000, and 10 to the power 9, which is a billion, is many hundreds of thousands. In fact, it's very close to a millionfold difference. Now we come to the real key where the control comes in. What then happens, and as biochemists have beautifully revealed for us over the last 40 or 50 years, a whole army of enzymes, those are chemicals that can facilitate reactions, which facilitate both the cutting of a nucleotide sequence and the pasting together of a nucleotide sequence. They come in and, directed by the cell to do the job, they discover where the errors are. You can do that, of course, because in practice what is happening is that they detect where there's a mismatch. The T is not connected to an A, and it has therefore to be corrected. So they actually go in and cut it out and put another one in. I mean, it sounds absolutely unbelievable. But yet, this is what we know. Chemically, that is what happens. Now I come to the control. You see, that process is what ensures that normally when a cell gets its DNA copied, there will be an almost exact copy in the daughter cells. In fact, the error rate then is only about 1 in 10 billion. That's a fantastic performance by nature, and we have to applaud whatever enabled this to develop, but it's what we find. Now we come to the interesting question. What was going on during the pandemic when we were invaded by coronavirus? In your body and mine and everybody else's body, something very interesting was happening. The organism's immune system had detected that a new invader had arrived. We did not have the immunoglobulin, that's the protein that is necessary for grabbing and holding on to the invader and neutralizing its action. But we did not have the right immunoglobulin to attack that the relevant immune system cells to stop the error correction being so completely accurate, so that naturally, by random errors in that initial stage of allowing the sort of crystallization, if you want to call it that, of the nucleotides to occur, so that a lot of errors were allowed to happen. That produces literally millions of new nucleotide sequences from which the immune system can choose. I use these words very deliberately. I know that some chemists and molecular biologists might bridle a little bit at the word choose, but I find it very difficult to use any other word. It literally says, okay, this cell has produced an immunoglobulin that grabs the virus. And, well, what do? We tell that cell to reproduce as fast as possible. And all the other cells are told to apoptose. That's the technical word for please die, please go. Now, I think the standard evolutionary biologists, meaning those who subscribe to the modern synthesis or neo-Darwinist synthesis, would say, well, Dennis, that's an exception. It's what Richard Dawkins will say. And that's an exception, Dennis. That's just the immune system playing around with a very clever mechanism. We agree. It's strange, but it is nevertheless not the usual situation. Well, I looked, largely prompted by a great biochemist at the University of Chicago, James Shapiro, the initiator of the concept of natural genetic engineering, the idea that the organism can, as it were, go in and change its genome. He certainly has shown that it's not just the immune system that does this. What he actually showed was that bacteria do it. When a colony of bacteria are under stress because of, for example, an antibiotic, they do exactly the same thing. As a colony, they allow their genomes to mutate and they reduce the error correction so that, just like the immune system, some of those in the colony will find a solution. And that's how we develop the resistance of bacteria to antibiotics, a big health problem, incidentally. But what he showed, in effect, was exactly the same process occurs in a colony of bacteria. Well, is it general? There's the remaining question. And through reading Shapiro's book from 2011, his book on evolution, I discovered, and you can all easily check it in the paper published in 2001 by the team that did the first sequencing of the complete sequencing of the human genome. And what that paper in 2001 shows in figure 42 of that paper, there's the detail, is extraordinary. They looked at a comparison in different organisms, all the way from microorganisms to the human, of particular kinds of proteins. The two proteins that they looked at were transcription factors and chromatin proteins. And what you find is extraordinary. As you go from the very simplest organisms to much more complex organisms, there is an accretion of domains. The proteins become longer with much more functionality, but they've not done so by the standard theory of evolution, which is tiny mutations slowly accumulating to produce a new sequence.